afternoon. Welcome to the 5th Annual EHE 360 Global Patient Conference. Thank you for making time to be here today. My name is Denise Robinson, and I am the Executive Director and Director of Research at the EHE Foundation. I want to start the day by thanking our event sponsors, Vivace Therapeutics and SpringWorks Therapeutics, who helped make this conference possible. And importantly, I want to thank the presenters and panelists who you'll hear from today and tomorrow. It's a two-day conference, as well as our moderator, David Casimir. I also want to take time to thank our volunteers who contributed to today's agenda and tomorrow. And those people make our community stronger, and they make this event stronger every year. If you are not familiar with the EHE Foundation, we are the leading global nonprofit advocacy and research organization dedicated to finding treatments and a cure for EHE. Everything we do aims to improve the lives of people impacted by EHE by advancing science and providing support. And we do that by funding impactful research, by facilitating education, creating disease awareness, building collaborations, and mobilizing a global community. These are central to our mission, and they wouldn't be possible without the support of donors and this community. We invite you today to ask questions throughout the event. It's a two-day event. Use the Q&A functions that are in your tools at the bottom of your screen. The community grows stronger through your sharing and your questions. And today's audience is filled with knowledge from patients, researchers, clinicians. So we want you to take advantage of that. I want to start today in recognition of the 10th anniversary of the EHE Foundation. We are pleased to have Dr. Brian Rubin as our keynote speaker. He's going to speak on reflecting on 10 years of discovery in EHE research and give future perspectives. Dr. Rubin is a renowned pathologist and chief of the Diagnostics Institute at the Cleveland Clinic. He is a longtime advisor to the foundation and the global EHE community, and truly a champion for EHE patients and all patients that he serves. So please welcome me, Dr. Rubin with me to the conference.
All right. Thanks very much, Denise, for the kind introduction. It is an honor and a pleasure to be here on the 10th anniversary of the EHE Foundation. I was there for the inauguration, the development of the foundation back many years ago, and remember, conversations with Jane Gutkiewicz very fondly about starting a foundation. And there was sort of one that already formed on Facebook page, and then we just kind of made it official. And so it's really nice to see what's happened with this foundation over the years and how it's grown and developed. It's really amazing. And today I'm just gonna talk a little bit, I'm going to reflect a little bit on the last 15 years of discovery and future perspective of EHE. And of course, the story of EHE is longer than that, but really the last 15 years is when I kind of got involved. And so it's a little bit of my personal journey and some observations I've had. So why don't we go ahead and get started? I will advance to the next slide. So, the story really begins, and there was a lot of groundwork before, but there was a landmark paper in 1982. I've showed this slide many, many times. These are two people I really look up to. They are essentially the father of soft tissue pathology, Franz Enziger on the left, and then his pupil, Sharon Weiss, who was a mentor to me my entire career. She's now retired, but two really great pathologists. And they worked at a time when they were describing different entities, and you can see them here actively looking at something. Maybe it was an EHE case, and they published this very landmark paper in 1982 describing a novel vascular tumor called epithelioid hemangioendothelioma, and that was really the beginning. So let's look at this timeline that we put together here. So please don't be offended if you're not on this timeline and you feel like you've made an important contribution or your group has. I didn't systematically make this, but I pitched in with a couple other people to try to remember some of the real important things so we can talk about them that happened, especially since I got involved around 2011 with this story. But seeing the left side of the timeline there, 1982 is when Enzinger and Weiss described epithelial hemangioendothelioma for the first time. And then you can see this isn't really proportional. There's really a long period of time between 1982 and 2011 where not a lot happened. I mean, people recognized EHE. It was a really hard diagnosis to make for most pathologists, but not a lot happened. But then, what really did happen was next generation sequencing and our ability to really genetically interrogate and evaluate a lot of things in ways that just were unprecedented. And so we were the first group to actually use next generation sequencing to identify a gene fusion in something that had a karyotype and a known translocation. So we, in 2011, we published our finding of the WWTR1 CAMPTA1 gene fusion in EHE. And contemporaneously or simultaneously, Christina Antoniescu and her group at Sloan Kettering found the same thing. And actually, they used an entirely different methodology of finding the genes. We published our work relatively at the same time. And it was really a validation that this was really a true finding. And what we found was that this gene fusion was found in the vast majority of EHE, about 90% in our combined studies. Also, at that time, there was a group in Australia, the International Herd Support Group, and they published their findings actually with Guy Weinberg, a longtime supporter of EHE. And so there was a lot going on at that time. And the Herd Group, essentially, from what I know, I was never involved with them and I never actually saw the data, but essentially it was a patient group that got together to share clinical findings related to EHE. And then Guy got involved because he was more of a scientist and helped them to publish. So 2011, big year for EHE. In 2013, an important result was that Christina Antoniescu and her group at Sloan Kettering published an influential paper showing that there was a related vascular tumor that had a YAP1 TFE3 fusion. Some people view it as a subset of EHE. Other people view it as a distinct entity. It doesn't really matter. It came out of the EHE group. But that happened in 2013. Then, of course, moving to 2015, we've got 3 foundations kind of coming together. And I think there was a lot of activity in different countries. But what was nice about this whole EHE work was that people started to form an international group of foundations. And I think that's very, very important here in the US, the UK, and over in Australia as well. From a scientific standpoint, that year was the year that a Japanese group actually came up with the idea of using CAMPTA1 immunohistochemistry as a marker for diagnosing an EHE. And that was important because this is a immunohistochemical marker. It's a stain on a glass slide, and it's a surrogate for the molecular gene fusion. And actually, it's a really good surrogate. I was just using it on a case this morning that I was looking at. And so that was an important development because it allowed everybody to sort of make this diagnosis of EHE with some certainty because of CAMPTA1 immunohistochemistry. It really helped the field a lot. In 2016, I think you're going to hear from Munir Tanas today. Munir was a postdoc in my lab at the time. And actually, Munir was the lead author on our 2011 paper with identification of the gene fusion. But then he extended his work. And, back in the early days, we really saw the need to develop biological models. People kept on asking me, what can we do to push this field forward now that you've got the genes? And I said, well, we need biological models. And we put a lot of effort. It seems like we spent most of the last, 15 years developing these biological models. But we did, Munir persevered in the lab and developed what we call a heterologous model in mouse cells, essentially mouse fibroblast cells, but we were able to express the gene fusion and learn a lot about it. And we published that work in 2016. And what was important about that work was actually we identified TEAD as a potential target. Even in the early days, we thought it was going to be a good target theoretically, but it was nice to see it in action. And we published that paper, I believe, in Oncogene back in 2016. In 2017, well, we knew TEAD was going to be a good target, but we actually put a lot of work into what could we harness early on and repurpose drugs that were already available to try to our lab that was actually published later in clinical cancer research led to a phase 2 trial with trametinib and EHE, and we did that work with Scott Shusey, was the PI from University of Michigan. We actually have published that work now as well in clinical cancer research. We saw a nice signal, but trametinib was a rough drug, and a lot of people left the study just because they couldn't tolerate the drug. But a lot of patients did stabilize on the drug, and so we know that the target of trametinib, which is a MAP kinase pathway, is an essential SIG transduction pathway within EHE. And we're going to try to leverage that information later on in later studies, perhaps in combination. This was also the first year that Guy Weinberg started this YAP-TAZ and TEAD meeting in Telluride each year. Cravat Foundation, which Guy founded to study EHE and other vascular cancers, was able to raise money through some connections. They made this small, very nice meeting. About 20 people attend each year. Guy funds the registration. Everybody pays their own way, essentially, to get to this meeting. And we made a lot of really important connections with people in related fields and in industry during those early years. And that meeting continues today. We were just discussing it's going to happen again in June. And John Lamar, who you'll hear from today, co-hosts that meeting with Guy each year, and I'm sure it'll be a good meeting. I'm unfortunately not going to be able to attend this year, but I know it will be a really good meeting. There'll be a lot of interactions, and I'm sure some very interesting work will come out of that. Now, in 2019, an important paper was published using foundation medicine data, so publicly available data. The colleagues out of Ohio State published this paper looking at secondary genetic alterations. And I think we've discussed these things before, but what we mean here is that TAS-CAMTA1 or WW1-CAMTA1 is the main driver of most EHE. But in addition to that, what we see is in aggressive EHE, we start to see secondary genetic alterations. The most common would be loss of a tumor suppressor gene known as CDKN2A. And that is seen in aggressive EHE. And so we started to learn a bit about genomic progression, which is a huge problem. We still sort of have a better understanding today, but it's still something that's really on my list of things to understand better. Now, 21. So there's a lot going on during these times we're talking about now to develop a mouse model, a genetically engineered mouse model of EHE. That mouse came to fruition. We published the first paper in 2021. So what we did was we took the WW1-CAMTA1 gene fusion, and using genetic engineering, we engineered it into the germline genome, so into the testes and ovaries of mice. And it allowed us to breed mice that would develop EHE. And it has actually pretty high penetrance. About half the mice will develop EHE, and we've credentialed that. It looks just like human EHE at every level. Clinically, it affects the same organs. Histologically, it looks the same. If you do immunohistochemistry, it's the same. If you do sophisticated molecular analysis, looking at gene transcription and things like that, it looks the same. And so this has really been a really strong model of EHE. About that time, we started to see phase 1 studies of TEAD inhibitors. So people have asked me before, were you the one who got people to start studying and developing drugs for TEAD? And the answer is no. TEAD had been on the to-do list for pharma for years and years and years as a really nice target for all sorts of cancer. It just turns out that EHE is addicted to TEAD through this WW1-CAMTA1 fusion, which that fusion needs to bind TEAD in order for the cells to work, tumor cells. And so TEAD is an elegant target. But these trials were done with other cancers in mind for the most part. The most common one would be mesothelioma, which is driven by a similar process to EHE. However, we are able to put EHE patients on these trials. So the first phase 1s were happening. at Vivace, at Akena, Novartis now, and other groups are getting into the game. There's many, many TEAD inhibitors in the clinical pipeline. There was a nice pair, Silvia Stocchiati out of Italy, got together a consensus group, a really huge group of people to talk about how we diagnose and manage EHE. That was published. Consensus guidelines are always really important in every field. Sort of thing. Hang on one second. I see this little button here. Okay. Every time I click a button, I hope it doesn't do anything negative, but I'm still here. And then in 22, we developed cell lines. And so what we used was our genetically engineered mouse model, a variation of it essentially that had lost CDKN2A to start generating cell lines because there were no human lines really available at this point. And really, for all practical purposes, we still don't have a human cell line. We've got a xenograft that came out of Italy, but and that's that EHE PDX, a patient-derived xenograft in Italy. And they have some cells that grow from that, but not really a full-on line yet. So human cell lines are still really a big need. But we do have this unlimited number of mouse cell lines that we can produce now. We have 3 that are very stable that we use for a lot of work, and we're developing even some new ones with different genetic backgrounds. But this mouse, one of the purposes of the mouse model was to develop a lot of tumors, so we would have a lot of ability to develop cell lines. Cell lines are nice because you can do all sorts of things to them in a plastic dish to try to understand therapies, biology, all sorts of things. Now, also in 22, we established EHE biobanks, both in the US and the UK. The US one is here at the Cleveland Clinic, and I don't manage it, but I actually look at all the cases and credential them and whatnot. But I think that work is going quite well. Biobanks are extremely important so that researchers from all over the world can get access to tissues and other biological things that are related to EHE patients. In 23, the EHE Global Patient Registry was started. So this is a patient kind of driven registry, recording all the characteristics of EHE patients all over the world so that, again, we can really refine our understanding of the clinical aspects of EHE. It's related to the biobank. So a lot of the patients, even in the registry, are members of the biobank as well, if you will. In 2024, so using those cell lines that we developed in 22, Ajay Pabani from my lab published a nice study looking at drug repurposing break. Essentially very active drug. There's not really a great clinical drug available right now, but there's a lot in trials. And CDK9 inhibition could be something nice to work in combination with TEAD inhibitors or even alone, and we're looking at that. And now here we are in 2025, and we've got multiple TEADs in phase 1 trials, and it seems like there's a lot of plans to be launching even more phase 1 trials. So let's go ahead and go to the next slide. But that's really in a nutshell. And I hope, I know that we still have patients dying of EHE every year. I lose friends, really. I've made a lot of friends through this group, and people I know that have had EHE for years to this terrible disease. But I would say, look at all this activity, look at what we've accomplished. And, I'll get into it in a second, but I think the future is very promising for therapies to treat this disease. So, again, where are we? So, from what I just talked about, we understand the genetic basis of this disease as well as genetic mechanism of disease progression. That is a lot of work happened by a lot of different laboratories to get there, and really important stuff. We've got validated EHE biological models. You always heard me screaming about biological models in the early days. We have them now. We still need human lines, but that is something that we're working on. Everybody in the field's working on it, and I believe we will be successful in developing human lines as well. We've got an EHE biobank and patient registry. These are really important foundational things to have for any disease that people want to study. Got the foundations. The EHE Foundation's been absolutely critical for funding research, for helping patients. through the Telluride meeting and through our attendance at more broad meetings. For instance, I go to the Hippo Pathway meetings every year. John Lamar does. Others who are now EHE researchers go to these other meetings. I would say that the scientific community has taken note. EHE people are very interested in this in different fields. For instance, the Hippo pathway, and EHE is related to the Hippo pathway because WWTR1 is one of the end effect way. And so more researchers are joining our ranks and looking to study EHE. Finally, we are advancing the treatment of EHE in multiple clinical trials are happening. Please support them. What are the gaps? So really interested in more reliable ways to follow EHE to monitor for early progression. That's going to be really essential, is how we can learn to recognize when patients are going to progress before it happens clinically, because that's our window of opportunity for therapy. Again, I mentioned human EHE cell lines. We need some models of the YAP1 TFE3 version of EHE. We don't have those really yet. We've got some heterologous models, but we need mouse models. We need some human cell lines. That's a real gap. And what we really need is a variety of effective drugs to treat EHE. Have come so far. I know we couldn't have come this far so fast without a lot of people involved. And so I want to thank the foundations, the other researchers, everybody who's been involved in this story. It's been a tremendously successful story. That said, we've got a long way to go. We did this by having a vision, supporting each other, staying consistent, and staying persistent. Those are very, very important. And I really, again, want to thank the community for just sticking with the research wing of this effort and continuing to support us. That has been absolutely critical. Now, we still have a way to go, so we need to maintain our focus, continue what we have been doing. Final message is don't give up. I do think that effective treatments are coming. They will be available, FDA approved at some point in the near future, and then we will really start to count our successes. Well, it's available for everybody. Okay, I think that's it. I wanted to thank everybody. I'm going to stop sharing.
Thank you, Dr. Rubin. That was awesome. Thanks for going through the timeline with us. And it really is great to see how far we've come.